This is Charter California Edition. My name is Brad Pomerance, and we are joined by Jennifer Armstrong. She's a professor at Scripps College, Claire McKenna College, and Pitzer College. How are you a professor at three colleges? Explain.
I will. I'm happy to. I'm a professor at 3 of the 5 Claremont undergraduate colleges because I'm a member of the Keck Science Department.
Right.
And our department is an interdisciplinary department of biologists, chemists, physicists, environmental analysis, neuroscientists, and we serve 3 of the 5 Claremont colleges.
And I wanted to have you join us today because we're continuing our conversation about the STEM fields. We have continuous conversations about building STEM in our great state. And given that Scripps is also a college focused and exclusively for women, it's nice to see that women continue to grow in the STEM fields. I want to speak about your work. It deals with fruit flies,
yes.
And you are using those flies to energize, to engage your students. Explain.
Well, there's so many wonderful things to talk about.
Yes.
So my laboratory has been very fortunate to be funded by the National Science Foundation, and the NSF funds our work to understand how basic biology happens. How do genes know when to be turned on? How are genes regulated in the cells? How are the structure of chromosomes maintained?
But here's a simple question for a simple
yes.
man like me. Fruit flies don't really look like humans.
No.
They're not chimpanzees or apes, and I'm not going to take a position on studying those animals, but explain how the study of fruit flies has complete applicability to you and I.
That's a great question. So NSF, as well as the NIH, funds work on these basic organisms that we call model organisms.
Yes.
And so there's fruit flies, there's worms, there's single-celled organisms like the yeast we use to make bread.
Right.
And the way that these organisms do their biology, they divide their cells, express their genes, is really the same way that we do.
Which is stunning to me on so many levels, to think that little yeast cell is just like any cell in our body.
So now that we can do a lot of genomics, we can ask these questions of how similar are they? And of genes that cause cancers when mutated in humans, about 2/3 of those genes are present in fruit flies. So we can examine what are they normally doing in the fruit fly to understand what's going wrong in us when they're mutated that might lead to cancer.
And what's stunning when you look at this very question is that you've been a professor for only 12 years. You're relatively young, and yet your undergraduate students are able to do experiments you couldn't have even done back when you were getting your doctorate or postdoc.
Oh, absolutely. So my undergraduate students are doing these experiments that are so sophisticated and so complicated. They're using machinery, equipment that didn't exist when I got my PhD. They're doing experiments that are so novel and so new that when they graduate, I have to run into the lab before they leave and say, teach me your protocol that you developed, because I need to know.
What are they studying right now?
So right now we're working on this protein that's called CHD1 or CHUD1. And I became interested in this protein a few years ago during my postdoc at UC Santa Cruz. We found that it was located at every active gene. If there's a gene and it's being expressed in a tissue, this protein CHUD1 is there. And it's a machine. It's a little machine that uses ATP to do things.
ATP is?
ATP is the energy currency of the cell.
Got it.
So instead of $1, you use ATP.
Well stated.
So we wanted to know, why is it there? What is this little machine doing at these active genes? And so we began first by asking, well, what would happen in a fruit fly if the fruit fly didn't have this protein? So my undergraduate students set about using genetic tools to make a mutation. They mutated the gene and asked, what happened to the fly?
And one would think that with a protein as dramatic and important as the one you just described, the fruit fly would not reproduce, would not survive.
And that's what we predicted. We predicted it would be dead, and maybe we would study what happened before it died. Because a lot of other genes that are related to the gene we study, the fly is gone, is dead when they're mutated. But it was so not dead. It was such a surprise.
Not dead.
It was not dead. The fly was alive. It was sterile. It had these interesting defects in their wings.
Why sterile? It seems like that's a common occurrence when there's a problem. What is that?
We don't know why they're sterile. We've sent some flies over to a colleague at Harvard Medical School, and he was interested in looking at that. We don't know. So many things are happening during gametogenesis, and there's so many genes that are being reprogrammed that it seems to be an important step in development.
But it seems like, aside from just this incident, and maybe I'm above our pay grade, but why is it when there is a problem with a gene, sterility seems to be one of the results?
I think just because so many things have to go right in order for fertility to happen.
Interesting.
And it's easy to have something go wrong.
Right.
Whereas once you're developed as an organism, you can kind of
Right.
hang out for a while.
So when you turned off this gene,
Yes.
you found out.
We found out the flies were sterile.
Right.
They had these interesting wing defects. But the surprising thing was when we looked at their chromosomes and looked at whether or not genes were expressed, the genes seemed to be expressed. So this was a puzzle. What is our protein doing? And this work was done over the last 10 years, 12
Right.
years in my lab at Scripps. Scripps students were involved in this, Claremont McKenna students, Pitzer students. And what has come out since then and from the work of other people
Sure.
is that this gene, when mutated in humans, is associated with prostate cancer. And nobody knows why. Nobody knows why prostate cancer.
So we can have the very mutation you inserted into the fruit fly.
Yes. So at the
Were
level...
you aware of that when you did this 10 years ago?
No. We were just curious. Why
Right.
is it there? It was basic biology. What does a gene need to do in order to be expressed?
Let me ask you, and forgive me, how do you mutate it? How does that happen?
Oh my goodness, that's a whole nother
do you do
story.
that?
There's a lot of different ways, a lot of different tools you can
Okay.
do. What my students did at Scripps, at Keck Science,
Yeah.
was that they used a technique that's called a jumping DNA.
Okay.
So there's these jumping genes
Uh-huh.
that were discovered in fruit flies, and we have them as well.
Okay.
They're very disturbing. They jump around
Okay.
and just hop around our genome, and we have remnants of them throughout our
Sure.
genomes. And so we used one of these jumping genes to jump out, and as it jumped out, it took some of our gene with it, the CHUD1 gene.
I'm blown away. I'm that guy who's blown away.
And this was done by undergraduates. They did it by crossing fruit flies,
Can you
analyzing the
see
DNA.
it?
You can analyze the DNA and using techniques like PCR, which is what they use at like CSI and things, to say like, oh look, there's our mutation. Our gene was mutated. Let's look at the fly. It's alive. It's sterile. It's got these interesting wing defects. Let's look at its chromosomes. When we looked at the chromosomes, we got an even bigger surprise. The chromosomes were like
Funky?
strange and funky. Yeah. So it seems to be important for global chromosome structure, and that's what we're following up on now.
What about lifespan?
Oh, that's such a great question.
Yeah.
My student did something about stress. She was interested in could they be stressed, and they
Right.
seem to be a little bit sensitive, but we haven't checked lifespan. We should do that. It's a
Oh,
very
see,
good
maybe
idea.
So the key question is, why is this beneficial for your students?
Yes,
Why
it's a
are
great
you
question.
such a fan of this work?
Because doing this work, it's fascinating,
Yeah.
but it's really hard. It requires, it's technically hard. These experiments are technically very challenging. You're moving tiny amounts of clear liquids from tube to tube. It all has to be done
Right.
perfectly. It's intellectually very challenging. Designing these experiments is hard. Figuring
Attention
out the
to
right
detail.
controls, attention to detail, interpreting the data when it comes out not quite what you had expected. It's just very exciting work, and the skills that you learn doing research are applicable to so many fields. So our students go on to graduate programs to get a PhD in biology at Johns Hopkins, at Harvard Medical School, University of
Right.
Colorado, Fred Hutchinson Research Cancer
Right.
Center, a few of my students.
Right.
But they also go on to medical school. They could become doctors. They become
Right.
genetic counselors. They could become high school teachers. They learn critical thinking. They learn how to really wrestle with data. They learn how to think clearly and how to be self-critical, because it's one thing to have a great idea about, I think the protein does this and I'm going to design experiments to show it, and it's another thing to think, what if that's not right? What if I'm wrong? And so you really have to be your own worst critic and examine your preconceptions and examine your assumptions.
I am so glad you came on, because you continue to show us how important the STEM fields are. And it's really an inspiration to see that our young men and women are getting excited about something like fruit flies and see how it applies to our human experience.
Absolutely.
I want to come to your lab.
Please, anytime.
Okay. Her name is Jennifer Armstrong. She is a professor at Scripps, Claremont McKenna, and Pitzer College. I'm Brad Pomerance. You're watching Charter California Edition.